Okay, so welcome back to this next video in which we are discussing circadian clocks. Okay, right, so we're currently in the process of discussing the mammalian circadian timing system. Okay, and we've discussed the suprachiasmatic nuclei, which are these two nuclei in the anterior hypothalamus, okay, which are positioned directly above the optic chiasm here. Okay, and these nuclei each consist of around 20,000 neurons in humans, okay, and we've discussed that all of them have this transcriptional translational feedback loop involving period and cryptochrome proteins perfectly in synchrony with one another. And we've said that even though the mechanisms by which that perfect synchrony is achieved are not well defined, it's certainly easily imaginable that they could keep each other all in synchrony, basically. Okay, so they're all going to have this transcriptional translational feedback loop in synchrony with one another. We've then discussed how the suprachiasmatic nuclei are kept in synchrony with the light-dark cycle, because not only are they all in synchrony with one another, but we want them all to be then in synchrony with the light-dark cycle. Okay, and we've discussed that there is this connection between these special photoreceptive ganglion cells in the retina and the suprachiasmatic nuclei, okay, which can then deliver information about the light-dark cycle to the suprachiasmatic nuclei so that they keep their circadian oscillators in synchrony with the light-dark cycle, i.e. with period and cryptochrome low at midnight and high at midday. Okay, right, so now what we want to discuss is what does the suprachiasmatic nuclei actually do, or rather, what do the suprachiasmatic nuclei actually do? Okay, so they are the master clocks. They control many different things. Now, before I actually move on to that great question of how they synchronize so many other peripheral cells with the light-dark cycle, what I firstly want to talk about is the function of the suprachiasmatic nuclei in regulating body temperature and also the sleep-wake cycle. And then I'll talk about the regulation of cortisol levels and melatonin levels in the blood, okay, and potentially how cortisol levels and melatonin levels in the blood then communicate to all other cells in the body and tell them what the circadian time should be so that they can keep their circadian oscillators in synchrony with the light-dark cycles. Okay, right, so let's start off by discussing the regulation of body temperature. Now, the first thing then that we should discuss if we're going to discuss the functions of the suprachiasmatic nuclei are that the rate at which they fire action potentials also shows circadian oscillation. Okay, so what I mean by that is probably best shown with one of these graphs. Okay, so once again we'll have time on the x-axis here, and that will go up in jumps of 4 hours, so 0, 4, 8, 12, 16, 20, 24, okay, and on the y-axis this time then, we're going to have the firing rate of the neurons in the suprachiasmatic nuclei, okay, so how often they're firing action potentials. And basically you find that they fire action potentials lowest at midnight, okay, and then the rate at which they fire action potentials goes up to the maximum at midday, and then it will drop back down to the low level for midnight, basically. So again, another circadian oscillation, and we can see that it's in phase basically with the levels of period and cryptochrome within the suprachiasmatic nuclei neurons. Okay, right, so this is the firing rate, how often they're firing action potentials, basically, the number of action potentials that they will fire in a certain amount of time. Firing rate. Okay, right, so why is a question you might have. Why are these neurons firing more action potentials during midday and less action potentials during night? Well, one of the explanations might be to do with the levels of a certain potassium channel. Okay, so there is a special potassium channel that I just want to introduce you to, which is known as the BK Ca. Okay, and you might put a little 2 plus there. Okay, so what is this then? This is the big conductance calcium activated potassium channel. Okay, so the B is for big conductance. The Ca2 plus down here is for calcium activated, and then the K is for potassium channel. Okay, and some people will just call these the big conductance potassium channels. Okay, now these are potassium channels that can be opened by intracellular calcium, basically. They can also actually be opened by depolarization of the electrical potential difference across the cell membrane. So really they're actually both calcium activated and voltage activated potassium channels. Okay, and the big conductance is because they have a large conductance for potassium ions once they are actually open, so their ability to let potassium ions move through them is very, very good, basically. They will move a lot of potassium ions once they're open. Okay, now, basically, the expression level of these big conductance calcium-activated potassium channels on the cell membranes of the suprachiasmatic nuclei neurons also shows a circadian oscillation, and we know how the expression levels of proteins can show circadian oscillations. Okay, we know that the BMAL1, NPAS2/clock heterodimers are very powerful transcription factors. Okay, and those their activity is oscillating on a 24-hour basis, and therefore the genes that they control will also be oscillating on a 24-hour basis. Okay, so the big conductance calcium-activated potassium channel basically is one of these genes that is having its expression oscillating on a 24-hour basis downstream of our initial transcriptional translational feedback loop, basically. Okay, now, it turns out that the big conductance calcium-activated potassium channel goes up during midnight, and then it goes down to a minimum at midday. Okay, so if we were to plot the expression level of this potassium channel, it looks like this. It goes down to a bottom at midnight, and it rises back up to a maximum it goes down to a bottom at midday, and then it rises to a peak at midnight. Okay, so this is the expression level of our big conductance calcium-activated potassium channel here, our BKCA2+. Okay, so basically these suprachiasmatic nuclei neurons express very high levels of the big conductance calcium-activated potassium channel at midnight during the night, and then very low levels during day. Okay, now why could this explain this firing rate graph here? Well, basically, if I draw a little picture of the big conductance calcium-activated potassium channel, this is a channel that will be open from time to time, and which will be allowing a lot of potassium to leave the inside of the cell and go to the outside of the cell. Okay, so when you open a potassium channel in the cell membrane, usually what happens is potassium then moves out of the cell because there is a much higher potassium concentration intracellularly. Okay, so normally in cells, the concentration of potassium intracellularly is 155 millimolar, whereas the concentration of potassium extracellularly is 4 millimolar. So that's around a 40-fold gradient favoring the movement out. Okay, now if you want to decide whether an ion, or rather which direction an ion is going to move in when you open a channel, you can't just look at the concentration gradient, you have to also look at the electrical gradient. Okay, now usually the resting electrical potential difference across a cell membrane is around negative 65 millivolts, meaning that the electrical potential intracellularly is 65 millivolts lower than the electrical potential extracellularly. Okay, now potassium ions are a positively charged ion. They do want to go where the electrical potential is lower, and at present that is the intracellular compartment. So actually the electrical gradient says the opposite thing to the concentration gradient. The concentration gradient says go out. the electrical gradient says come in. The question then, of course, is who wins? Who is stronger? Okay, and it turns out that normally the concentration gradient of 40-fold is stronger than negative 65 millivolts, basically. Okay, so potassium ions will move out normally at that electrical potential difference. That is not strong enough to stop potassium moving out. It will weaken it, of course. You won't get as many potassium ions leaving as you would if you didn't have that electrical potential difference across the cell membrane, but it's not big enough to stop it. Okay, so you will get potassium ions moving out. So basically, if we are putting these big conductance calcium-activated potassium channels into the cell membrane during night, okay, and taking them away during day, then at night what's going to be happening? We're going to have new potassium currents leaving the cell, okay, that weren't there before. Now what's this going to do? Well, it means we're moving potassium ions from the intracellular compartment to the extracellular compartment. Potassium is a positively charged ion. Where you put positively charged ions, you increase the electrical potential. Okay, so what are we going to be doing to the extracellular and intracellular electrical potential? Well, if I just denote the extracellular electrical potential as capital E for electrical potential, and then subscripted E for extracellular, and I denote the electrical potential intracellularly as capital E for electrical potential, and then subscripted I for intracellularly. When we dump potassium ions into the extracellular compartment, we're going to be raising the electrical potential in the extracellular compartment. Okay, because after all, we're dumping positive charge there, so we raise the electrical potential extracellularly. Okay, when we remove positive charge from the intracellular compartment, we're going to be reducing the electrical potential of the intracellular compartment. Now, what's that going to do to the electrical potential difference across the cell membrane? Well, previously we could represent it like this. It's helpful to represent it with a simple picture like this. This is the electrical potential extracellularly, and this is the electrical potential intracellularly. Before we started moving the potassium ions, there was already a drop of negative 65 millivolts. Okay, the intracellular electrical potential was lower than the extracellular electrical potential by negative 65 millivolts, and that was the electrical potential difference across the cell membrane. We have now raised the electrical potential extracellularly, so this line's been moved up. We are now reducing the electrical potential intracellularly. This line's being moved down. What are we going to do now to the drop down? We are going to make the drop bigger, basically. Okay, this number, the electrical potential difference across the cell membrane, will become more negative. Okay, and therefore the electrical potential difference across the cell membrane is going to become hyperpolarized. The exact opposite of what you need to do if you want to fire an action potential. Okay, so at night we will be hyperpolarizing the electrical potential difference across the cell membrane of the suprachiasmatic nuclei neurons, and that will make it much more difficult for these suprachiasmatic nuclei neurons to fire action potentials, and that could explain why the firing rate of these suprachiasmatic nuclei neurons is decreasing during the night, and then why it's higher during the day. Okay, so to summarize, because of the transcriptional translational feedback loop, because of our circadian clock basically, which we know changes gene expression within cells on a 24-hour basis, one of the genes that is being regulated on this 24-hour basis is the big conductance calcium-activated potassium channel subunits. Okay, and they have a low expression during the daytime and high expression during the nighttime. Okay, so you're going to have more big conductance calcium-activated potassium channels during the evening, during night. Okay, and that's going to be present within your cell membranes of your suprachiasmatic nuclei neurons. Okay, that means that during the night you're going to be hyperpolarizing the electrical potential difference across the cell membrane of your suprachiasmatic nuclei neurons, and that's why they fire at lower rates during the night than in the day. Okay, right, so the suprachiasmatic nuclei neurons then are firing at a higher rate during the day and at a lower rate during the night. Okay, now the suprachiasmatic nuclei neurons connect to many other portions of the brain and then affect their activity. Okay, so because of this rhythmical firing where they're firing at higher rates during the day, okay, they're now going to be activating or potentially inhibiting other portions of the brain to differing degrees depending on the time of day. Okay, and this is going to underlie the different effects of the suprachiasmatic nuclei. Okay, so the first thing I want to talk about is body temperature. Okay, right, so firstly let me just explain to you that body temperature does vary on a circadian basis, basically. So body temperature is another example of a circadian rhythm. It cycles with a period of 24 hours. Okay, now what we'll then talk about is how the suprachiasmatic nuclei are going to regulate this. Okay, so firstly let me draw you another one of these graphs then. So on the x-axis we'll have time again, so here 0, 4, 8, 12, 16, 20, 24, okay, and now on the y-axis we'll have body temperature. Okay, now body temperature then can vary between a low point of around 36.7, okay, and then up to a maximum of 37.1. So body temperature doesn't vary that much. It stays around 37, okay, but at its minimum it can go down to 36.7, and then at its maximum it can go up to 37.1, okay, and basically it's at its lowest point during the night. So body temperature is at 36.7 during the middle of the night, and then towards midday body temperature will be growing to the highest level, okay, and then it will fall back down during the afternoon and evening till it gets back to that lowest level by midnight. Okay, so that is the circadian oscillation of body temperature. It's at around 36.7 at midnight, and it's at its maximum of 37.1 during midday, basically. Okay, right, so body temperature is an example of a circadian rhythm. Okay, now why? What on earth causes that? Well, the answer is we don't totally know. However, we can link the suprachiasmatic nucleus, or rather suprachiasmatic nuclei, to another hypothalamic nucleus, or hypothalamic nuclei, which control body temperature. Okay, and this is one of those hypothalamic nuclei that we went over when we looked at the anatomy. Okay, so basically the anterior hypothalamic nuclei are responsible for controlling body temperature. Okay, so I'll just get back up my pictures of the hypothalami. Okay, and we can see the anterior hypothalamic nuclei again. So remember, these are these ones here. They're sitting basically behind the suprachiasmatic nuclei. Okay, remember in our first cross section we were right at the front here, and there were the suprachiasmatic nuclei. Now if you go back a little bit, okay, you now see the anterior hypothalamic nuclei in that equivalent position. So basically these are sitting behind the suprachiasmatic nuclei. So the suprachiasmatic nuclei are very close to the anterior hypothalamic nuclei. Okay, so they have neural connections with the anterior hypothalamic nuclei. Okay, and during the day they will be giving higher firing rates to the anterior hypothalamic nuclei, and somehow that higher rate of input from the suprachiasmatic nuclei drives the anterior hypothalamic nuclei to raise body temperature from 36.7 as it is at midnight to 37.1 as it is at midday, basically. Okay, right, so by communicating to that other hypothalamic nucleus, or nuclei rather, the suprachiasmatic nuclei are controlling body temperature, the circadian rhythm of body temperature. Okay, right. Another really important thing to say, although I can't really give any more insight into this because it's just not understood at all, is that the suprachiasmatic nuclei are extremely important in controlling the sleep-wake cycle, the circadian regulation of the sleep-wake cycle. So they are extremely important in making sure that you are awake during the daytime and asleep during nighttime. Okay, if you remove the suprachiasmatic nuclei, okay, then animals just start sleeping in little chunks all over the place, basically. They no longer stay awake throughout the entire day and then sleep through the entire night. Instead, what happens is they will sleep for 2 hours, then they'll get up for 2 hours, sleep for 2 hours, get up for 2 hours. They'll end up doing that, basically. Okay, so the suprachiasmatic nuclei are extremely important in the sleep-wake cycle, basically. But we don't really understand sleep-wake cycles at all. Okay, so I can't really tell you anything more about how the suprachiasmatic nuclei control the sleep-wake cycles.